In order for replication to begin, an enzyme called DNA helicase must bind to the origin of replication and break the hydrogen bonds that hold the two complementary strands of DNA together. This forms two Y-shaped replication forks that together form a replication bubble. As helicase unzips the DNA strands, an enzyme called gyrase attaches to the outside of the replication fork to alleviate stress and prevent supercoiling of DNA strands, and single-stranded binding proteins move in and attach to the separated strands of DNA to prevent them binding back to one another. Once the replication fork has been created, an enzyme called primase makes a short piece of RNA called a primer. This binds to the 3' ends of the strands. The primer is necessary for polymerase III to bind in order to replicate the DNA. Polymerase III replicates the DNA strands by reading the strands in the 3' to 5' direction and making complementary base pairs in the 5' to 3' direction. As polymerase III replicates the strands, helicase continues to unzip the strands and grow the replication bubble. Because DNA strands are complementary and anti-parallel, and new DNA strands are written in the 5' to 3' direction, as helicase continues to unzip the strands, it creates a situation where one strand can be written continuously, which is called the leading strand, and one strand lags behind and has to be created in several smaller fragments. This strand is called the lagging strand and is making up by several Okazaki fragments. These Okazaki fragments are made by RNA primase attaching near the replication fork and polymerase III writing the complementary fragments of DNA until it hits the primase that was laid from the fragment before. Because primase is not a functioning piece of the DNA code, it has to be written over in order for that replicated DNA to function. Polymerase I comes in and replaces the portions of DNA that primase had previously been attached to. Then an enzyme called ligase glues the fragments together to create a complete and functioning DNA strand. All of this is done in a complicated simultaneous manner.